Hello and welcome to this Neurotech investor webinar discussing today's announcement from the company on its phase 1/2 Rett Syndrome trial results. All participants are in listen-only mode. There'll be a presentation lasting for approximately 30 to 40 minutes, followed by Q&A. If you have a question you'd like to submit, please feel free to type it in using the Q&A function within Zoom. Please note this session is being recorded. On behalf of Neurotech today, we have the Executive Director, Dr. Thomas Duthie. I'll now hand it over to you. Please go ahead.
Thank you, Matt, and thanks everyone for joining. It's nice to reconnect on some additional clinical data we have for you today in Rett syndrome, having only spoken to, I assume, some of you a couple of weeks ago with the results of our phase 2/3 autism clinical trial in paediatric patients. So it's been a very, very busy month for the company, and we're delighted to share with you today the very exciting results we've seen today for our Rett syndrome phase 1/2 clinical trial. This is just a disclaimer. There will be forward-looking statements made today in this presentation that has been lodged with ASX. Just want to remind everyone before we get into the Rett data that we embarked on this four-core strategic approach at Neurotech on or around September 2022. And we more or less pivoted the business to focus exclusively on paediatric neurological patients. So in essence, we're following the evidence of NTI-164, which has been shown preclinically to have very strong anti-neuroinflammatory effects and neuroprotective effects in vitro. And so with that, we've been scouring for paediatric indications where there's a correlation between persistent or progressive neuroinflammation in these children. And we're focused on registering NTI-164 as a broad-spectrum cannabinoid drug therapy, hence why we're investing in these clinical trials and we're investing into our preclinical work. And we focus our efforts on partnering with key paediatric neurologists so far in the Australian context who've discharged all four of our clinical trials with utmost integrity. And we've been delighted at the rate at which we've been able to recruit patients, report data, and then continue to develop this really exciting asset. And that's reflected here in our pipeline. We now have a bona fide clinical development pipeline in 2024. And pleasing today that we now have the full results of our phase 1/2 trial in Rett syndrome. So that means since this strategy was essentially earmarked, notwithstanding some early work in autism, we've now more or less delivered statistical significance and clinically meaningful data in 3 separate paediatric neurological disorders. 2 of which we consider rare or orphan, that being Rett syndrome and pediatric acute neuropsychiatric syndrome, or PANS, and its close cousin PANDAS. So very pleasing from our perspective. Going now to Rett syndrome, I put this quote up last time. I think it sort of harnesses everything we try to do for these children as clinical and corporate developers of assets in Rett syndrome. These children historically have been very, very difficult to manage with no approved standards of treatment until March of 2023 with the approval of trofinetide DAYBUE for Rett syndrome, which is a drug developed by Neuropharmaceuticals. Many of you know that I'm unashamedly a fan of Neuropharmaceuticals and the work they've done to develop their asset. And so that they and their partner Arcadia are the first to market with trofinetide in the United States. This was our trial design. It was 14 girls. It was open label. So essentially the endpoints you will see today have been developed against baseline scores. So we use the patient's own baseline metrics when measuring the change or the difference at 12 weeks. And like with all of our trials to date, we up titrate the dose in the first week to find their, if you like, their tolerated dose. And then they receive that dose for a further 8 weeks. And in this case, we have a 12-week endpoint. We haven't seen, we have had all patients, as we've announced to ASX, extend to 52 weeks of daily oral therapy of NTI-164. And these patients all completed 12 weeks of daily treatment with NTI-164. The batteries of tests are standard tests with particular reference to CGI-I and RSBQ, which were co-primary endpoints for DAYBUE's trofinetide's registration study in the United States. And what we have as our format, format or drug offering, is NTI-164 which is derived from a cannabis strain. It is a broad-spectrum cannabinoid drug product, and we have shown, and we believe we have a good entourage effect where these cannabinoids synergize each other to potentiate a greater or enhanced effect in these patients. And I mentioned we have data to support the fact that this format is both neuroprotective and anti-neuroinflammatory. And we'll come to these secondary measures and, of course, the important primary measure shortly, which on a top-line basis was reported on April 17. These were our baseline patient characteristics for the trial. Probably no surprises that all patients were female girls, 8.8 mean age, weight 27.5 kilos, and baseline RSBQ score, which is important from our RSBQ measure, was 44.6, and our CGI-S mean score at baseline was 4.6, and we stratified which patients were deemed moderate, marked, or severe at baseline. This is our 12-week safety data, so probably no surprises here based on the evidence that we've had across both PANDAS, PANS, and autism, both in the short term over 4, 8, and 12 weeks, but also in autism's case, well over 90 weeks now of daily oral treatment. We haven't had a significant number of deleterious side effects here in this study. We did have one serious adverse event where one patient started developing a rash, urticaria, or hives. We've never seen that before, interestingly, across any patient that we've treated to date, and that's sort of close to 94, 95 children now. And the adverse events for this trial were not only well tolerated, but certainly very manageable. So 11 AEs across 4 patients. For weight loss or weight gain, we didn't see any change from baseline, albeit when I say that, there was a 300-gram difference, so slightly more weight put on at 12 weeks versus baseline, but for purposes of this disclosure and this presentation, largely unchanged. Vomiting, we had 2 patients record a vomiting event, 14%, and we saw no diarrhea. So with respect to the diarrhea and the vomiting, very, very consistent, or lack of diarrhea and vomiting, very, very consistent with what we've shown in those previous trials. Their bloods are also normal, no reportable events there. And we have provided some context there with the registration study for debutyrofenatide and the AE rates for that format. So in conclusion, for our safety, we show an excellent safety profile, and we show minimum or minimal patient-specific side effects. So fantastic result on safety. With efficacy, this is again a little bit of a tortured slide here with a few pieces of data. I'll go through at least 3 of these in more detail in coming slides, but I wanted to make the observation here for people on the call today that the 2 most important measures from the perspective of registering a drug for Rett syndrome in the United States and elsewhere is CGI-I and RSBQ. For CGI-I, and I'll come to this, we saw overall an improvement of 10% from baseline at 12 weeks when we looked at 9 exploratory Rett-specific anchors. And I today put into this deck and also our ASX release that this was a proof of concept trial. So Associate Professor Carolyn Ellaway picked 9 Rett anchors, not knowing whether NTO-164 would impart an effect or not on any one of those 9. And what we now have is that we were able to elicit a very strong response in 4 core Rett anchors specifically, which we would intend to use as we further develop the asset. And that is not inconsistent with all previous Rett syndrome clinical trials. You find the domains you want to prosecute, and they're the ones you stick with. So if we look at those 4 key Rett anchors for us, we saw a 23% improvement in CGI-I at 12 weeks with a 0.1% likelihood that that was by chance alone. Similarly with RSBQ, again, this is all 14 patients delivered RSBQ at 12 weeks. We saw a 13.4 score decrease, which is an improvement of 30% and less than 0.1% chance that was a fluke. So statistically very strong. Are they clinically meaningful? Yes, we strongly support the fact they are clinically meaningful differences. And then our other secondary measures, you can see for the majority of these measures that we used, we also did see an improvement and that improvement was statistically significant. So again, I know I said this last time, this is 14 patients. It is an open label study. There is no placebo. It is not blinded clearly, but to exert an effect on 14 girls when your secondary endpoints are not powered for significance, and to see significance highlights to us that we are seeing consistency of the improvement across different assessments, and particularly those that relate to communication anxiety, socialization, and alertiveness. Associate Professor Allaway has established the Rett Syndrome Multidisciplinary Management Clinic in Sydney at Westmead Children's Hospital. And she's really hit the nail on the head, I guess, with the quote today, looking at what's important to her as a doctor treating Rett syndrome patients, but also, I guess, most importantly for caregivers, the parents of these girls. They really want these children to see improvements in the way they communicate, improvements in some of their repetitive hand behaviours. Certainly anxiety and mood is important. And overall, of course, you want to be able to improve these girls' quality of life. And we've done that in this small phase 1/2 clinical trial. And we have done so without compromising these patients' safety, with a very, very manageable and a low number of adverse events as they relate to vomiting. We had some quite brief caregiver testimonials around how they've seen a change in their daughter's life, if you like. I guess for people on the call today who are otherwise healthy, these seem incredibly incremental, but for a patient and their parents, these are somewhat transformational differences living day to day in that household. So again, that should be caregiver number 4 there at the bottom. I apologise for that. These are separate patients and separate parents. CGI-I, this is the primary endpoint. So on the 17th of April, on our top line disclosure, which was just this top line data point you see here today, we had 4 Rett-specific anchors to report on that top line, and that was the mean difference at 12 weeks of 0.3, with a confidence interval as described. Just a reminder, the 95% confidence interval is basically a statistical measure that if I was to repeat, or we were to repeat this trial 100 times, 95 times out of 100, the true effect would lie somewhere between those two numbers, so 0.015 or 0.56. So on May 6th, which is the data today, we now have the fulsome analysis of the CGI-I endpoint, where all of our Rett-specific anchors, so for all 9 of them, and they're shown there on the right-hand side, were available for analysis and interpretation. And on the whole, we did see a slight increase in the improvement relative to the top-line data, and a slightly better p-value. This is the patient-specific responses of the 9 Rett-specific anchors. So again, the all-in, if you like, CGI-I score at 12 weeks. And we saw that essentially for week 12 data, you can see we've broken out week 4 as well. No change in 43% of patients, 50% were minimally improved, and 1 patient was minimally worse. So that's for all 9 anchors. But as I mentioned, as this is a first-in-human clinical trial of 164 in Rett, we wanted to have as many anchors in there as possible to see what effects, if any, NTI-164 was having on these 9 different Rett-specific anchors. Would have been amazing if we conferred a huge benefit on each of the 9 anchors. That would be a truly remarkable drug intervention. But we did not see equal effects across all 9. But we did see really strong effects in 4 assessments that are reported here on this slide, which I'll come to. And interestingly, they correlate with the kinds of improvements that I mentioned and is contained in Carolyn's quote that doctors and caregivers see as important. And those 4 for us are communication skills, mental alertness, socialization, and anxiety. And you can see if you just look at those 4 domains, not looking at any other of the 5, you see that the CGI-I score improved by almost 1 scale point, and it was statistically quite strong. Moreover, if you look at the individual patients, so the 14 girls that were administered these 4 domains, bearing in mind all 14 girls got 9 domains, I've just carved out the 4 domains here, you can see that there's a profound effect on their improvement. So in this case, when you just focus on those 4 domains, only 1 patient was unchanged clinically. 57 were minimally improved, 29 were much improved, and 1 patient was very much improved. So in essence, looking at these 4 core domains, 93% of our patients improved as it relates to CGI-I. Turning now to RSBQ, which is the key secondary endpoint. So RSBQ is a very standard, validated FDA measure, EMA measure that assesses the severity of the neurobehavioral problems from the perspective of the caregiver. And it is, as I mentioned, one of the most widely used measures in Rett syndrome due to its excellent inter-rater and test-retest validity. And of course, it's accepted by the FDA for use in Rett syndrome trials. Obviously, without having a placebo here, we can compare the RSBQ scores only to the baseline measure. So the baseline measure for all patients was 44.6 at the mean, and at 4 weeks, we started to see an improvement. So that improvement at 4 weeks was minus 4.4 on the Rett Syndrome Behavioural Questionnaire score. That's not a bad improvement after 4 weeks. By week 12, we saw a 205% improvement in their RSBQ scores, negative being an improvement, but from 4.4 to 13.4. So this is a very strong change out to week 12. And what it means for us is that when you look at the data more carefully, so the RSBQ composite score is what I've just talked about. Then we have on the right-hand side of the slide the subdomains. So these are the subdomains that are additive to give you the overall score. So you can see here that we were really good at hitting mood, hands, facial expressions, body rocking, and fear and anxiety. We didn't there was possible trend, if you want to call it that, in breathing, nighttime activities and walking, standing. But again, these were not statistically significant, so no difference really from a purely statistical metric at 12 weeks. When you overlay that result with the CGI-I scores and those domains I mentioned, coupled with some of the other measures that we captured in this clinical trial, I hope I can convince those on the call that we now know a lot about the kinds of effects we can see in these children as they relate to not just Rett, but most importantly as well, all 3 of these indications, because there's a lot of consistency here on these domains in autism and PANS as well. So if anything, NTI-164 has shown really commendable, repetitive, beneficial effects in these disorders, which gives us great encouragement, not only on the development of this asset, but also how we can move this asset into other neurological or rare neurological disorders of children where there's a neuroinflammatory effect. One of the other secondary measures is CGI-S that we looked at. So again, clinical global impression severity of illness. It's a 7-point scale, 1 being normal, 7 being extremely ill. We were able to shift that CGI-S score by 0.4 of a scale point overall at 12 weeks, and statistically that was significant. We also see an interesting effect with our CGI-S in other trials. And as our patients now are moving or have moved into the 52-week extension, we'll continue to collect not only this CGI-S data, but also CGI-I, RSBQ, and other. And interestingly, from an autism and PANS perspective, I'm not trying to overreach here and link it to Rett, but we have seen historically continued improvement to 24 weeks and 32 weeks for PANS and autism respectively. So it will be interesting from our perspective as we continue to watch these girls to see whether we can continue to improve them on both CGI-S and CGI-I. I wanted to just frame one slide here to show you 2 things. Firstly, I wanted to just highlight here that the 2 graphical pieces you're looking at, this is not in the context of the same trial. And so it's just for very basic comparison purposes only between the data that we have by way of trofinetide's registration and the data we have today in our phase 1/2 open label study of NTI-164. So essentially it's just lining them up. There are different statistical measures used. Obviously these are different patients, US-centric Australian patients, But I wanted to highlight here that overall, we're very pleased with the percent improvement in our RSBQ score from baseline to 12 weeks. And notwithstanding the fact we didn't have a placebo arm, if there was a placebo effect as measured and seen in Neuron's registration study, I think we still, if that placebo effect was replicated in a hypothetical sense, we still have, I guess, plenty of wiggle room here on our RSBQ score. And again, the benefits we're seeing in RSBQ and CGI-I are not coming at the cost of significant patient morbidities and side effects. So this is an important aspect for us, and one that clearly we are going to look at developing in Rett syndrome based on RSBQ and CGI-I. There were a bunch of other secondary measures here. So they're acronym heavy, unfortunately, but again, I don't plan on going into these into substantial detail. They are assessment tools that are used for Rett. They're assessment tools that are used for children. ICNDS is the Impact of Childhood Neurological Disability Scale. And then it's relative, looking at just quality of life. They were statistically significant. The caregiver burden, or the Rett-specific caregiver burden index, was statistically significant. RTT-DSC-VAS basically is what's called a domain-specific concerns visual acuity visual analog scale. Excuse me. And there's 4 pieces to that. You don't sort of bang them together for a total composite score, that you report them separately. And as you kind of hope, we were able to see a communication benefit there for RTT-DSC-VAS. So in conclusion, I think we have demonstrated here as our first in man, first in girl, as it turns out, trial for NTL164, that we've shown a statistically significant clinical meaningful improvement in CGI-I with a mean change of 0.4, looking at all domains. But when we look at the 4 domains of interest to us as we develop the asset now, we showed a 23% improvement at 12 weeks, which is highly significant. And importantly, 93% of those patients, when looking at just those 4 core domains, were improved. The secondary endpoints, yep, the majority were met. The most important secondary endpoint is RSBQ for us. And we showed a statistically significant and clinical meaningful improvement in RSBQ. And as I mentioned, the difference between 4 weeks and 12 weeks on the change in RSBQ was quite strong at 205% improvement. We did have one SAE during the trial. We had a very small number of AEs relating wholeheartedly to vomiting. A couple of children, one got a common cold, one had pharyngitis, one had a chest infection, I think, with the other AEs. But none of these AEs were considered to interfere in the patient's functioning, and none of the adverse events, i.e. the vomiting one here that we called out, required any kind of anti-vomiting medication. So they were transient, in other words. We still believe, although there is a standard of care now debuting in the United States, we still believe there is always room in a market for more than one drug therapy. And there is always a need, no matter how good the drug therapy, for additional safe and effective new therapies over time. So I'd like to then just conclude here with our sort of milestone outlook. Again, the company has delivered in spades. We have had 3 clinical trials in pandas, PANS, autism, and now Rett syndrome, all basically deliver results, and we're very proud of that. In the second half, we have some more work to do. Obviously, we will continue to collect both Rett syndrome open label data, so we will be able to continue to report some measures like RSBQ and CGI-I, CGI-S, as we've been doing with pandas and PANS and autism. And we have some further data coming in our PANS study relating to the genotyping of these patients and how NTL164 may impart or how it imparts improvement on certain gene regulation. And I think the second half of this year is really there's quite a lot of activity. So we do have aspirations to secure an ODD for Rett syndrome in the United States and Europe, as we do for pandas, PANS, both rare orphan disorders. Associate Professor Carolyn Ellaway at this early stage is intending to present our work at an upcoming international Rett syndrome meeting, interestingly in Australia. We have some IND enabling IND enabling and EMA enabling toxicology work to complete. And we also expect to commence a cerebral palsy phase 1/2 trial. That's it from me. I would now like to turn the call back to Maddie for Q&A. Thanks, Matt.
Yeah, thanks, Tom. As you mentioned, we'll move on to the Q&A. The questions are based on some sent through via email and also live using Zoom. So if you have a question you'd like to submit, please feel free to type it in and submit it using Zoom. The first question, Tom, is how importantly do you view the safety aspects of NTI 164 relative to the efficacy at this juncture?
I would say that in all drug development, you always pay a price for efficacy, and that price you pay is typically safety. So it's not unusual to have some safety issues. It would be amazing if we didn't have any side effects, but we do. So I would say, with particular reference to NTI 164 in Rett syndrome, the clinical effects observed relative to the safety impact or impost on these patients is very strong. So there is a very strong thesis, clearly, to continue the development of NTI 164 because it's safe. And the fact that it's also clinically very efficacious, albeit caveated by the fact it's a 14-patient study, gives us, really strong grounds to continue our work with Associate Professor Ellaway and our work with global regulators.
Thank you. There's been a couple of questions come through on this, but you mentioned that there were 4 CGI anchors, namely communication, mental alertness, socialization, and anxiety, each of which showed a benefit, and they formed the basis for your further development. What were the anchor effects, and what were the CGI anchors used for trofinetide approval with Acadia and Neuron?
So we reported the 4 anchors in more detail than the other 5 anchors. So they were our best anchors that are the most prospective. The other anchors, so for example, we did see some improvements in the other anchors as well, but we didn't go into the minutiae of what those anchors specifically showed. That can form part of a future scientific paper or clinical disclosure at a conference, perhaps. But what I can say today is that we didn't really shift the dial at all on autonomic function. So this is the autonomic nerve function in these children is compromised. We didn't see any improvement there, really. They didn't get any worse. We didn't see any improvement in autonomic function. And we weren't able to impact seizure activity and sleep patterns. So, clearly the information on those is really important for us, because it would be a complete own goal for us to then take those 3 where we showed no improvement and use them as our 3 anchors in, say, a pivotal registration-directed trial. And this is all permissible. This is exactly what you do when you're developing a neurological intervention, and with particular reference to Rett. There was, I think, a company called Anavex who had tens upon tens of different anchors they were looking at initially to try to work out how they could target their drug on particular domains and which ones it was efficacious in. And Anavex subsequently didn't work. Phase 3 failed in January of this year. But we know, based on the clinician advice, is that you take 2 to 3 of these anchors into your registration trial, CGI-I, and that's exactly what we've done. I think the second part of the question was, do we know what the Neuron CGI anchors were, the Rett-specific anchors? There's certainly reference literature around the kinds of anchors that are important in Rett, but we ourselves at Neurotech, based on some literature searches that we've undertaken, we don't know, and we haven't seen what those anchors were for CGI-I as their primary endpoint, and how many there were. So it's a bit unknown to us, to be frank. If it is published and someone's got it, I'd certainly like to see it, because it might make for some further interesting dialogue, particularly if Neuron are hitting some of those similar domains that we are. And I think that answers both parts of that question, Matt.
Yes, thank you. The next question is, what additional data can we expect from the extension phase of the phase 1/2 trial?
Yeah, I covered that in part. The beauty of running proof of concept open label trials is they're not only cost and time effective, but they give you an incredible amount of information like we've seen here today. There's so much data that we've generated here. It's really exciting. And, we've assimilated as a company our data set that is most important for the development of NTI-164. But of course, there's a heap of other data that we will work through in the coming weeks. The beauty of having an open label extension, and then it's well flagged because Neuron's done the same with its LILAC trial and post-Lavender approval. You can also continue to develop the data, but also you can continue to report the data. So for us, the way we think about this is that these girls are being monitored, they're being measured, and they're being assessed. And so what we should see over time is exactly what we've been able to do with our other indications, and that is at 24 weeks, 30, 52 weeks, give or take. Don't hold me to those exact numbers, but we will have points in time where these girls will be measured and monitored, and we'll be able to report that data as well. As I mentioned on the call, historically, we continue to see improvements beyond 8, 12 weeks of initial treatment, depending on the trial. So it can only and forever be helpful for these patients, clearly, if they don't stagnate, and stagnate's not the right word because they've improved significantly from their baseline. But if they don't improve any further from 12 weeks and we hold them into that clinical status, if you like, I think that's a commendable outcome, particularly given the safety. And I'll just very quickly overlay something here. In all our studies to date, we have seen no rebound in symptoms. So if the patients stay on treatment and they don't fall off, they may not get any better after a certain period of time, but they certainly don't get worse. And that's been also a very clear message. As long as you take the intervention, you will receive the benefit of that intervention in terms of your clinical symptoms.
Thank you. The next question is, what are the next steps for Rett syndrome development?
The next steps are, as I flagged, we are looking to secure orphan drug designation in the United States and Europe for Rett. And we are also currently thinking about exactly what that next trial looks like, how that looks from a registration perspective of NTI-164 in certain markets. Obviously, this is day one of the data that we've got, but on the strength of this data, we are fully intending, as best we can, with our regulatory engagement strategy and our clinical engagement with Associate Professor Carolyn Ellaway, to really hone in on what we've shown here today to design a very smart clinical trial that will deliver statistically significant and clinically meaningful differences to allow us to then potentially move forward with a full commercial plan for NTI-164 in Rett syndrome. But as I've said on previous calls, we have taken somewhat of a different strategy than most. We've actually stuck to our balance sheet principles of being a small company without significant access to funds, and we have run these trials in Australia. So, the next step here is to involve international key opinion leaders and experts on top of those unbelievably passionate and supportive key opinion leaders we have in Australia, and that's Associate Professor Carolyn Ellaway for Rett syndrome. It's Professor Russell Dale for PANDAS-PANS, and it's Professor Michael Fay for autism, and Professor Michael Fay sort of sits across the other two trials. So it's been a good journey, but it's only just begun.
Next question is, are there any other rare pediatric neurological disorders that you're looking at?
We've got Human Research Ethics Committee clearance for cerebral palsy. There is some evidence of persistent neuroinflammatory effects in CP patients that Prof. Fay has published on. Again, we're very evidence driven, and so in short, the answer is absolutely yes. There are a number of rare pediatric neurological disorders where they have abrogated or disrupted pathways, and those pathways can lead and do lead in some indications to what is the core principle of every study we've done, and that is it leads to neuroinflammation. So with this data, with PANS and Rett, we have not disclosed beyond cerebral palsy what else we're interested in. But as we develop the literature and speak to our key opinion leaders and convene advisory boards, I can't help but feel excited that we have really strong opportunities in the future to look at certain other pediatric neurological disorders where there's a significant unmet need and there's a significant opportunity for NTI-164 and collectively Neurotech.
Tom, can you comment on the various secondary measures outside of RSBQ, which you have shown, and how they compare with the debut phase 3 clinical trial?
Right. So there were some common, and it's apples for oranges, and everyone on this call knows how much I respect John Pilcher and the work that Neuron's done and the team at Larry Glass. I've known them for a very long time. It's not about what they've got and what we've got, but the common sort of secondary measures beyond those ones that I've mentioned was I have to look, sorry, down on my page. RTT-CBI was one that they looked at. INCDS, quality of life, ICNDS also were in common. CSBS, DPI-T, they looked at. We did not look at that measure. Professor Elway excluded it principally because our girls were too old, and it was more an infant checklist. So there was certainly some overlap. We showed some significance in some of those measures, as I mentioned. Neurons showed some significance in some measures and showed in others they were not significant. So I guess there's a blend here, but they weren't sort of equally identical per se, but certainly there was some commonality in that, and I'll leave it at that.
Thank you. The next question is, given the autism results were outstanding and it has such a large unmet need, why is Neurotech pursuing other indications?
That is an amazing question because I've just delivered Rett syndrome. Well, the company has just delivered Rett syndrome data, which looks highly prospective, as we have done for PANDAS and PANS. So for autism, let me take that in a slightly different way. Whilst it is admirable and effective for companies to be wholeheartedly tunnel and razor-like focused on one disease or disorder, the fact is, for autism, it is not an orphan indication, and autism will require 2 well-designed phase 3 clinical trials to register that drug in Europe and the United States. I've mentioned previously, as it relates to autism, that on the basis of our 54-patient phase 2/3 trial, we do, and that trial being undertaken in Australia, we do plan to sit down with the Therapeutic Goods Administration to understand whether that clinical trial or not is acceptable to consider provisional style applications or not, or what other form of clinical data that the TGA may or may not require from Neurotech. The reason we're going and looking at the TGA first is exactly what I just said. The company is not funded to complete 2 pivotal phase 3 clinical trials, well, even 1 phase 3 clinical trial in the United States, if our first one counts, which we don't know in the United States context yet, so we haven't had the discussion, but you do typically require 2 phase 3s. And so for us, with the NDIS chewing $7 billion in taxpayer dollars with 235,000 level 2 and 3 autistic children on that trial, on the NDIS, we can't help but think the best opportunity for autism is in Australia. Now, is it cost prohibitive to do that? No, it is not. But we do have to sit down with the Therapeutic Goods Administration to map that out. However, for Rett and PANS, as Neuron has demonstrated with Arcadia, one well-designed phase 3 clinical trial with two co-primary endpoints of CGI-I and RSBQ. If they're met, that is registration worthy, as we know now. And so for me, why not continue to develop those assets where the timelines are compressed to market, the cost is compressed, and also the regulatory levers you get from looking at rare orphan disorders, and more particularly rare orphan pediatric disorders. They give you so much more assistance than you otherwise might get for, say, adult disorders or disorders where the prevalence or incidence is quite high. So I'm unwavering in my view that the company does need to have multiple shots on goal and does need to find the fastest way to get NTI-164 to market. And that is going to come from a TGA strategy in Australia, initially for autism, and it's going to come from a more globally focused strategy for pandas, PANS, and Rett. But are we interested in Australia for both of those indications? Of course we are.
Thank you. Given the excellent results and assuming orphan drug designation, can you outline what is likely for the next clinical trial and how Neurotech plans to finance that?
I think from the perspective of Rett syndrome, it's too early to call out the costs associated with a trial that we haven't exactly costed, and the protocol is not developed, and the sample size is nascent at best. What I would say though is that on the basis of say our autism trial, which is 54 children in the Australian context, at least it was less than about a $3 million investment. This Neuron trial is less than $1 million. This Rett's trial for Neurotech is less than $1 million of Neurotech investment. So these are not huge costs relative to the opportunity. And so I probably can't answer that yet, except to say we will plan a more sizable registration trial for Rett moving forward, as we would, of course, for PANS.
Thank you. From a practitioner perspective, how do these results position NTI-164 versus DAYBUE? Are these results compelling enough, if validated in a subsequent trial, to take a share or potential first-line therapy from DAYBUE?
So to take the last bit first, I think this was sort of asked in a different way last call, but essentially in the United States, DAYBUE trofinetide is a standard of care. It is the first and only approved treatment. I think at this stage, my view is that for any developer from here on end, if they want to contest against DAYBUE in the United States, they have to either do one of two things. They have to combine with it to show that when you put the two together, you see an additional benefit that's not at the cost of safety. Or they would need to show at some point that having one treatment versus DAYBUE is superior. We look at it from the perspective in the United States that patients will start on DAYBUE. And so for us, that market dynamic is different. For us in the United States, it's a if you like, a cleaner market in Australia because DAYBUE is not approved here. So we are able to run placebo. We will be able to run placebo-controlled trials presumably here. Now, that's not to say that ultimately in the United States there's no market opportunity. Of course there is, and I think I made mention of this on the call last time that for patients who are unable to tolerate DAYBUE or their symptoms return and they are required to step off DAYBUE therapy, there's no reason over time to suggest that an intervention like NTI-164 or any other intervention may pick up those patients that, as I say, didn't respond to or dropped off treatment with DAYBUE. And that's called a second-line treatment. Are the results good enough? Well, we haven't got a placebo. We've run 14 patients, but as we know from at least our autism trial, that when we kind of supersize the sample size and blind it, we more or less replicate what we see in these open label extensions. And so I think today sitting here looking at those CGI-I domains, those 4 domains that are important for us right now, that is a very strong result, given 93% of children improved and only 1 showed no change. None got worse. From an RSBQ perspective, a 13.4 scale change is clearly meaningful, and as is a 4.9 change delivered by Neuron, because they were approved. That is meaningful. It was meaningful against placebo, where it was close to 3 times better. We just don't have the placebo data. So I think for us, what I've seen today, and it's clearly what's in Associate Professor Ellaway's quote, is that we've seen enough to say to ourselves, and certainly for our patients who are extending for 52 weeks, that this is an intervention that must be developed.
Thank you. How far is Neurotech away from monetizing NTI-164, and what are the options being explored?
Well, as I think I called out last time on the call and call out repeatedly when I speak to investors, is that my job here is to build the asset and to build the value in the asset by discharging clinical trials and also positioning the company from a regulatory perspective and a patent perspective to be somewhat, as best you can, to be bulletproof. And that's very well grounded in precedent. And so I only look to my revered company Neuron to see that the asset value they've created, not just for Rett, for trofinetide, tripeptide IGF-1, but also their next generation drug across multiple pediatric indications, is exactly how you build that value. So in short, the way to monetize that value from here is to continue to build the asset value through strategic clinical trials, through clever regulatory engagement, through the award of good regulatory levers, but also that monetization can take the form of partnering, and that partnering can be by way of licensing, as evidenced by Neuron and Arcadia. That is a $1 billion license deal that Neuron has unbelievably well executed. The second way is to monetize the equity value of the company by way of a potential merger or takeover. None of which I will be drawn on, because we don't provide running commentary on how or what stage or when we're going to monetize the asset. But the monetization process, I'm very happy to share with you, because that is exactly what we've been doing with these trials and the regulatory strategies that we've put in place. That for me is how we build the asset, and then the monetization happens either in parallel with that or at some point into the future.
The next question is, can you outline any intention or plan to commercially leverage these results via a partnership, and what could that look like? Is anybody knocking?
Probably just answered that question, if I'm honest, on the last question, to say that if people were knocking, we wouldn't disclose they were anyway. So again, we don't provide running commentary on confidential commercial discussions of any form. So I guess the proof in the pudding is always and forever going to be through the ASX that we've completed a partnership or a transaction of some sort. Until that point in time, I can't comment on the question.
Thank you. And Tom, another one that's come through. Congratulations on the results. What does news flow and reporting look like for the 14 patients who have been put onto the 52-week extension phase?
Yep, so I think we covered that. Again, just to remind the audience that as an open label study, we will continue to collect data pertaining to both safety and efficacy. Efficacy noting our 2 major ones, RSBQ and CGI-I. And so what you will see here are at certain points in time, we will be able to update the market on progress of those children, as we've done for autism and PANS. Again, without being committed to a week or a day, for autism and PANS, we did, so for PANS, we did 12 and 24 weeks so far. For autism, I think we've done 4, 8, 20, 32, 52, and 90. So a little bit of a blend. There's no real magic to it. It's it will be disclosed when we've received the data, and I'm as interested as anyone else in whether we can continue to impart an improvement of these girls over time, as we have with those other pediatric neurological disorders.
A couple of questions just around IP and patents. Can you just make a comment on sort of how protectable that is?
Yeah, no, I'd be happy to. So we have, I always look beyond patents. So I will comment on patents, but I'll also reiterate that an ODD in the United States confers 7 years market exclusivity from the day you launch. And in Europe, an ODD confers 10 years market exclusivity. So these are government conferred monopoly positions that stops anyone ripping off trofinetide, irrespective of Neuren's patents, as it would equally groups attempting to rip off, replicate, or copy 164 if we were able to achieve ODDs in those indications. Now, the backup here is the patents. So we have 2 patent families relating to both composition. We believe it is the novel facets of this particular strain in terms of the mix of the different cannabinoids led by CBDA, which is actually like a pro or mother drug to CBD, but also some of these other cannabinoids that we have in our drug product. So we have composition claims pending for that, and we also have methods. So you have patents relating to claims relating to a method of treatment. So a method of treating, for example, Rett syndrome. So those 2 patent families are now through the initial PCT process, the Patent Cooperation Treaty process. So it's 18 months from provisional filing from memory. And we're now at the national phase for each of our jurisdictions. So they're starting to develop up nicely. Do we have an issued patent on 164 or a method patent, composition or methods? No, we do not at this juncture. The other levers we can rely on are the fact that we control the process from essentially seed to patient. So we have the strain, we have developed the strain, we have a very strong and detailed understanding on how to reproducibly, with fantastic continuity of composition, how to grow it. So how to produce the strain that then delivers the replicable cannabinoid content we need. So the old batch to batch variation we've nutted out. It's been a very long process to get that right. And again, that's a trade secret of the company. And it's also kind of stacked on top of the patents, if you like, if granted. So there's sort of like a 3-tier approach here to harnessing, protecting, and nurturing the IP.
Thanks, Tom. The final question I've got is, has the Australian government offered or provided any funding or development support, and is there any pathway to securing such funding or support?
From the perspective of an Australian government grant or granting agency like the NHMRC and ARC for academic researchers, I guess, the short answer is no. But the Australian government provides Neurotech and a lot of our peers with the best incentive to invest into the clinical development and even preclinical development, and that is something called the R&D tax incentive. So for a gross outlay of $6.5 million in R&D expense in FY23, Neurotech was able to receive 3.2 million as an R&D tax incentive, and that was at or around the 48.5%, not the 43.5%, but the 48.5% level. Again, tactically, that's remunerative for us because we get close to half of every dollar we spend back in cash. And the reason it's high is because we have a lot of onshore activity here. We manufacture here, we do our preclinical work here, we do our clinical work here. So the mix is highly appropriate for, a large rebate, non-dilutive rebate relative to our investment overall. So again, as the company grows and our aspirations move to a more global type setting over time, then of course, so too does the benefits we've received to date. The R&D tax incentive might be slightly diluted.
Thanks, Tom, and thanks to everyone for the questions. Tom, I'll just hand it back to you to provide a closing comment before we end the session.
Okay, thanks, Matt. Well, thanks everyone for joining again. I know it can be somewhat torturous going through quite scientific data, but I really wanted to convey to everyone on the call today that despite this being a little bit heavy on science, you need to walk away from the call knowing that science is very strong. And I'd like to thank Associate Professor Ellaway and our contract research organisation for the diligence that they have displayed in making that trial happen for us, and the endpoints and the design that we attached to that trial, I think, have been nothing other than super helpful for us as we develop this asset. So again, we're really delighted that every child who's on our trial has stayed on the therapy. And as a corporate, we will continue to provide that therapy free of charge to every participant in any of our trials into perpetuity at the patient or caregiver's request. So thanks, Matt, and thanks everyone. And if you have any further questions, please feel free to reach out. I'm always available, sometimes a little bit slower on busy days, but certainly very happy to work with investors as we articulate this really exciting story. So thanks, Matt. I'll leave it there.
Thanks, Tom. Thanks again to everyone for joining.